Cysteine is the only universally affected and disfavored proteomic amino acid under oxidative conditions in animals. PY 2024 IR95 IR96 Germany.
德国
Cysteine is the only universally affected and disfavored proteomic amino acid under oxidative conditions in animals. PY 2024 IR95 IR96 Germany.
德国
Cysteine is the only universally affected and disfavored proteomic amino acid under oxidative conditions in animals. This article belongs to the special issue Oxidative Stress and Antioxidants in Computational Chemistry. Abstract: oxidative modifications of amino acid side chains and proteins are a hallmark of oxidative stress, and they are usually regarded as structural damage. However, amino acid oxidation may also have a protective effect and may serve regulatory or structural purposes. Here, we have attempted to characterize the global redox role of the 20 proteinogenic amino acids in animals by analyzing their usage frequency in 5 plausible evolutionary paradigms of increased oxidative burden. I. Peroxisomal proteins versus all proteins. 2. Mitochondrial proteins versus all proteins. 3. Mitochondrially encoded respiratory chain proteins versus all mitochondrial proteins. IV proteins from long-lived animals versus those from short-lived animals. And V proteins from aerobic, free-living animals versus those from facultatively anaerobic animals. We have found that avoidance of cysteine in the oxidative condition was the most pronounced and significant variation in the majority of comparisons. Beyond this preeminent pattern, only local signals were observed, primarily increases in methionine and glutamine as well as decreases in serine and proline. Hence, certain types of cysteine oxidation appear to enforce its proteome-wide evolutionary avoidance despite its essential role in disulfide bond formation and metal ligation. The susceptibility to oxidation of all other amino acids appears to be generally unproblematic, and sometimes advantageous. Keywords: aging, free radical, 1-electron oxidation, peroxidation, protein oxidation, thiol radical 1. Introduction: chemical modifications of biological macromolecules are important challenges to cellular and organismal fitness that are involved in numerous diseases and in the biological aging process. 1, 2, 3. In general, biological macromolecular persistence may be achieved by different strategies, among them reduced numbers or levels of offenders to structural integrity, such as oxidants, intrinsically enhanced stability, due to altered usage of structural building blocks, or improved mechanisms of replacement and repair, such as reducing enzymes, chaperones, etc. Regarding the structural stability of proteins to chemical oxidation, 2 opposing effects need to be separated: amino acid oxidation with overall adverse consequences, and amino acid oxidation with overall beneficial consequences. The former may arise when a major or minor route of oxidation leads to irreparable damage of the protein or its surroundings, such as during protein cysteine thiol radical formation 5, 6, which appears to account for the avoidance of mitochondrial membrane cysteines in long-lived animal species. 7, 8, 9. Cysteine thiol radicals can relay damage into the interior of proteins 10, and act as chain transfer catalysts in vivo 5. They are incompletely scavenged by aqueous antioxidants like ascorbate and glutathione 5, 6, 11. And no efficient system seems to exist for their scavenging in hydrophobic environments 6, 12. Thus, cysteine 1-electron oxidation or a related type of oxidation may account at least in part for the very low degree of conservation of solitary surface cysteine residues, as opposed to paired cysteine residues, in the general proteome. 13, 14. Since disulfides are much more resistant to 1-electron oxidation and less damaging in the 1-electron oxidized state than free thiol groups. 6, 11, 15. In contrast, other forms of amino acid oxidation may have systemically beneficial consequences, especially when the individually effectuated chemical modification is structurally rather harmless and, preferentially, readily repairable. Under these conditions, the oxidative event may have prevented potentially much more dangerous chemical damage elsewhere in the cell. For example, protein methionine oxidation to methionine sulfoxide can be repaired enzymatically. 16, 17, and induces only moderate structural deterioration 16, 18, 19, but effectively scavenges reactive species that might have triggered a more deleterious reaction cascade elsewhere. For example, respiratory chain complexes have been shown to profoundly accumulate this labile amino acid in parallel with metabolic rate and reactive oxygen species, ROS production. Clearly indicative of an adaptive antioxidant mechanism. 18, 20, 21, 22. In the present study, we have attempted to clarify in a systematic fashion whether the two well-established examples above, mitochondrial cysteine accumulation and mitochondrial methionine accumulation, indeed constituted the major proteomic amino acid adaptations towards oxidative stress in animals, or whether they just represented showcase examples of a much wider and diverse body of potentially adaptive, quantitative changes. Hence, a testing battery of 5 evolutionary paradigms of persistently increased oxidative burden in animals was devised, and the corresponding proteomes were comparatively evaluated for differences in amino acid usages. The 5 paradigms of comparison were designated peroxisome, I, mitochondrion 2 respiratory chain, 3 longevity IV, anaerobicity, V, and they relate to oxidative stress as follows. I. Peroxisomes have long been recognized for their high levels of hydrogen peroxide production, which locally and sometimes globally involves substantial oxidative stress despite high local concentrations of the antioxidant enzyme, catalase 23, 24, 2. Mitochondria usually dominate cellular oxidant production due to their potentially high superoxide release from flavoenzymes and ubiquinone oxidoreductases, 25, 26, 27, 3. The prime source of superoxide within mitochondria are the respiratory chain complexes of the inner mitochondrial membrane, 25, 26, which are by themselves in need of special antioxidant protection 18, 28, 5. Longevity requires particular antioxidant long-term strategies of prevention and repair, 1, 29, 30, several of which are realized through structural amino acid usage changes, 7, 9, 12, 20, 31, 32, 33, V, as opposed to anaerobic fermentation. Aerobic respiration involves cellular oxygen toxicity, which is mediated by the production of reactive oxygen species, ROS 34, 35. Through their regulatory shutdown of respiration, at least during adulthood, facultatively anaerobic parasitic helminths largely avoid oxidative stress despite the continued histological presence of mitochondria 7, 36, 37. Finally, to put the obtained animal data into a wider perspective, a collection of aerobic versus anaerobic prokaryotes with well-defined oxygen requirements was sampled and analyzed accordingly to materials and methods.